TropWATER James Cook University, in association with the Queensland Department of Environment and Science and Griffith University, are conducting a large research project funded by the Australian Government's National Environmental Science Program, Tropical Water Quality Hub, with additional support from the Queensland Government's Reef Water Quality Program. This program aims to characterise and trace the sediment as it moves from the paddock all the way out to the reef.
Our work involves sampling the end of river and also flood plumes. We carry out incubation tests in the lab to quantify the release of nutrients when fresh water mixes with seawater, and through microbial breakdown of sediment organic matter. We found that the release of nutrients from sediment can be considerable and that it varies between different flood events. And we also found that this is likely due to the type and source of organic matter transported with the sediment.
In addition, we've established several continuous monitoring locations of sediment traps and environmental loggers designed to monitor the sediment exposure and turbidity over 4 wet and 3 dry seasons.
There's 2 key instruments that we've used in the project. One is the improved sediment trap, and the other one is a high volume particle filtration system that's really proven itself.
We hope to use these data to better quantify the influence of the newly delivered sediment in the inshore Great Barrier Reef.
Once we get the samples back into the laboratory, there's quite an extensive process to prepare them for a range of analysis. Firstly, we need to remove the salts from the samples and then look at them under the microscope to identify the particles and how they are changing from the river and out into the marine environment. We conduct sediment grain size to determine the physical properties of the sediments, and we've also developed some improved methods for measuring the primary particles of these grains that are being preferentially transported within the Great Barrier Reef lagoon. After this process, we then sort the samples into the finer sediments, which are further analysed for geochemistry and clay mineralogy.
We have measured the bacteria and fungal community composition of the organic components that form the sediment flocks in river flood plumes. This research provides a more comprehensive understanding of how sediment changes as it is transported from the river catchments out to the marine environment.
Now that we have developed these new methodologies, we hope to apply these new methods to other river catchments of the Great Barrier Reef.
Our research will help prioritise remediation on the ground, and that will lead to healthier landscapes, healthier rivers and healthy reefs.